technical and conference papers. When it comes to achievements, he was the team lead of scientists who helped to develop over 100,000 mini tubers of yam for planting after the Kokumba-Nanumba War, I think somewhere in the middle of the 90s, around 94. Again, he was the manager of Bioplanters Ghana, and there he championed the production of over 1 million MD2 suckers for pineapple farmers in Ghana in 2004. He is currently a full-time lecturer at the Department of Nuclear Agric and Radiation Processing at the School of Nuclear and Allied Sciences. Ladies and gentlemen, with a round of applause, let's welcome our keynote speaker, Professor Amoatey.
I'll use this one. Thank you very much, Mr. Moderator. Good morning, directors. Good morning, scientists. Good morning, all of you colleagues I worked with some time ago. Glad to be here today. And after the introduction, I don't want to waste any more time, maybe a pleasantries to follow. So I'll go straight away to the presentation. Okay, so I'll do some small, my title of the presentation is Nuclear Science and Technology in Ghana's Agriculture and Food Systems. And this is a brief of the outline. I'll do some introduction and look at World War II and public perception of nuclear science, developments thereafter, IEA and when it was established, what it stands for, global applications of nuclear science and technology, impact of nuclear science and technology in Ghana's agriculture and food systems. And I'll also look at way forward, and we shall come to conclusions. I also took some time to put some abbreviations there to help us as we move along. Small definition before we move forward so that we are not lost completely. Several definitions will be found in the books, but I prefer the third one. Nuclear science is the study of subatomic particles and their application in various disciplines of human endeavor. I also like to give some small definition to radiation because we shall be talking quite a lot about radiation. And radiation is energy that moves from one place to another in a form that can be described as waves or particles. As humans, we are exposed to radiation in our everyday life. Some of the most familiar sources of radiation include the sun, microwave ovens, radios. Most of these radiations carry no risk to our health, while others do. And depending upon the type of radiation, different measures must be taken to protect our bodies and environment from its effects, while allowing us to benefit from its many applications. Mr. Moderator, I wish to beg your pardon and to inform the audience that I am aware that some of my slides are overloaded, but it is on purpose. One of which is that sometimes the information there is quite new to me, and I was unable to commit all to memory. Another is that sometimes the information there must be together so that we can flow. In others, it is scanty, and I also flash over. Thank you very much. So the science of atomic radiation, atomic change, and nuclear fusion was developed from 1895 to 1945, and much of it in the last 6 of those years. That's 1939 to 1945. It began with the discovery of X-rays in 1895 and ended in 1953 with the Atoms for Peace speech by a wonderful president of the United States of America. Like I said, this slide is overloaded, but I couldn't help it. So, but it's not too much for our presentation. So, most of the time when we are talking about nuclear energy and things like that, then people begin to say, oh, it's not good. That's the science that we don't want to hear about because you are preparing atomic bombs. Indeed, in 1945, the US bombed Japan, destroying 2 cities. The 2 events were the only times nuclear weapons were used in combat. And the effect is that 140,000 people were killed in Hiroshima and 70,000 in Nagasaki. Survivors contracted leukemia, cancer, and other terrible side effects. The photo I wanted to show is not too clear, so we shall move forward. So negative effects of nuclear science and technology, people fear that if you are dealing in this area, then you produce some waste by all means. And what do you do with the waste? So the waste can remain radioactive and dangerous to humans for several years. Fear from people is also that workers can be excessively exposed to nuclear radiation, which causes many, serious health problems. Following World War II, there was geopolitical tension between the United States and Soviet Union and their respective allies, purposely for global struggle for power and influence, and they started building nuclear weapons. The risk and why the whole world was against it, that nuclear weapons have potential to destroy the world's human population. This year marks exactly the 70th anniversary of President Eisenhower's Atom for Peace speech. And to commemorate this speech and the enduring impact of his vision which led to creation of the IEA, the agency and the United States of America hosted a panel discussion in Vienna at the IEA's conference, General Conference. President of Ghana, the U.S. Secretary of Energy, and the granddaughter of the former president of the U.S. joined the current director of the IEA on 3rd October to discuss the legacy of the speech and the impact of the IEA. So what is the IEA? The IEA is an autonomous intergovernmental organization dedicated to increasing contributions of atomic energy to world peace and well-being, and ensuring that agency assistance is not used for military purposes. The objective of IEA is to accelerate and enlarge contribution of atomic energy to peace, health, and prosperity. At the time IEA was established in 1957, it had 80 members. Today it has 178 members. The ultimate goal of IEA is to promote applications of nuclear science and technology for peace and development around the world for the benefit of all mankind. The IEA therefore initiates and maintains programs aimed at supporting tangible socio-economic development by promoting the use of appropriate nuclear science and technology in addressing major sustainable development priorities in the market. I pause here to define nuclear technology as any technology that involves the nuclear reactions of atomic nuclei. Globally, nuclear science and technologies are being used in various disciplines. This includes carbon dating, environmental monitoring, engineering projects, food and agriculture, human health, hydrological studies, industrial science, nuclear reactors for scientific research. I have food and agriculture because we shall be going there in details. Nuclear energy production, that is where Ghana, electricity production, I missed out. Ghana wants to be there soon. Because nobody is interested in that. Nuclear science and technology is also applied in smoke detection, space exploration, transport, waste management, and water desalination. Indeed, Ghana is doing some of them and not doing some of them yet. So now, Mr. Moderator, we are getting closer home now. And the role of agriculture. Before I get to the contribution of nuclear science and technology in Ghana's agriculture and food systems, we'll briefly go over the role of agriculture in the economy of Ghana. And I have indicated that it contributes to national income, it's a source of food for the population, provides raw materials for various industries, responsible for infrastructure growth, and enhances rural prosperity. Specifically, agriculture provides 20% of the food needs of Ghana, contributes 54% of Ghana's GDP, and employs 52% of the labor force, generates over 40% of export earnings. Documents vary with respect to these figures, but I got mine from FAO 21. So now we are going to look at 30 years of nuclear science and technology in Ghana's agriculture and food systems. Mainly research development activities and their effects on agriculture. And I'll focus on insect pest management, which is an important aspect of agricultural production in Ghana. Then I'll come to food processing and food irrigation in the context of preservation and food safety. Then I'll come to increasing food production, where I'll talk briefly about mutation breeding and biotechnology, and also soil, water, and nutrient management. Insects, most of them are beneficial and they pollinate our crops, also feed on other insects. However, a small number of them are major pests or vectors of major pathogens in livestock and humans. For decades, we have relied almost exclusively on insecticides as the main weapon for controlling the insect pests. But this has a number of challenges with respect to effect on human health, the environment, and insecticide resistance and residues in the environment. This calls for more environmentally friendly and sustainable insect pest control approaches. And sterile insect technique, SIT, stands tall when it comes to looking for alternatives. So the sterile insect technique is always all over the world used as part of an area-wide integrated pest control strategy to suppress, contain, locally eradicate, or prevent the invasion or reinvasion of insect pest populations and disease vectors worldwide. The method of application is very simple. It is used in ionizing, the SIT uses ionizing radiation from gamma rays or electron beam to sterilize mass-red insects, typically the male ones. The effect of the radiation is that there is chromosome breakage in the germ-line cells of the insect sperm. Lethal dominant mutations that prevent the development of embryos. So when these males mate with unirradiated females in the wild, in the bush, in our parks, no offspring are produced, and the result is that the sterility in the wild population and the reduction of pest population over time. Benefits of SIT. It is environmentally friendly compared to chemicals. It is specific because it targets a specific species and leaving all the others unharmed. It is effective. It's been used over 60 years in several countries all over the world. It is economical in territories where SIT was applied. Retrospective economic assessment studies have shown a very high return on investments. So, coming to Ghana, we have been interested in cheche fly because it is endemic in category areas in our country. Cheche fly is a vector of a tropical parasitic disease called trypanosomiasis. In some of the books, they refer to it as African trypanosomiasis, which I disagree with because trip is found in all tropical areas of the world. I don't see why they like to associate us with anything that is not good. So, cheche flies can harm cattle by causing a disease known as nagana. And the symptoms include intermittent fever accompanied by anemia, edema, lacrimation, and large lymph nodes, abortion, decreased fertility, loss of appetite and weight, eventually leading to death. It can also affect humans, and in humans it's called sleeping sickness. And the symptoms include sleeping during the day and inability to sleep at night. Irritation, confusion, unclear speech, headache, general feeling of unwellness, weakness, fatigue, fever, swelling of lymph nodes, liver, spleen, severe sweating. And then there is loss of weight, and there is eventual death. For people who may have lived in cattle growing areas, you would observe that the boys following the cattle are lean and emaciated, and the cattle are also lean and emaciated. This is not due to lack of nutrition. It is due to this sleeping sickness in the boys and nagana in the cattle. So, over the years, it was discovered through scientific research that the female cheche fly tends to mate with only one male in its lifetime. So, scientists have tried to exploit this. The procedure for the SIT is that you do mass rearing of the male insects and sterilize them using irradiation. And we said the exposure to radiation will make them sterile. Then you go into the field and release them. But the radiation should make sure that there will be no progeny. So the expected outcome of this mating is that no offspring are produced when such males are released in the field to mate with wild females. There is therefore a reduction of the pest population or complete eradication over time. How have we applied this in Ghana? In the late 80s to early 90s, some work was done between GAEC, Animal Research Institute, the Veterinary Services Department, with support from the International Atomic Energy Agency. And this led to reduction in prevalence of cattle production. Some work has also been done using the SIT on stem borer control. Once again, mass rearing was done, field releases of male stem borer was also done, but this time not over an extensive area, but in confined field trials. And this was in a partnership between Binari and MOFA under the Agricultural Sector Services Improvement Project. And the impact was elimination of maize stem borer in the area that was used for the controlled experiment. Currently, there's a global effort to use SIT for control of mosquitoes. So this is a global effort. Binary is part of this effort. Work in progress is, at the moment, studies on mass rearing and irradiation, and on field biology of the female. The partners are Binary and Ministry of Health. There is also ongoing work involving fruit flies. And at the moment, the scientists are working on the field biology of male fruit flies and on mass rearing and irradiation. Binary is the lead institute. During my preparations, I didn't get information on the partners. There is also work on armyworm which affects maize. Also, studies on field biology of the male armyworm, on mass rearing and irradiation. And I'm not sure of this Ministry of Health, which I put there myself. Okay, I think there's an area that the radiation entomologists can look at currently, and it should involve control of the coconut beetle. Ghana is currently rated 12 on the world ranking as coconut producer. And recently, more and more people are diverting from other crops to coconut. The effect of the coconut beetle is that it destroys the leaf, can eat up the whole core of the plant. We can see the destructions on the leaf. I'm trying to get this thing.
You want to go back?
No, I want to the officer will do it.
Laser.
The laser. Okay. Okay. Which also is the palm beetle, which is a delicacy in Ghana, especially in the rural areas. Mr. Moderator, I'm not advocating that we eliminate this from our environment, because the rural folks will be very angry with me, scientists, because they say I sit in Accra and eat processed food from Argentina or something. So we should find a way of protecting those ones while we eliminate the ones that are trying to block our progress in coconut production. I will move to food irradiation. Foods obtained from plant and animal sources begin to spoil soon after harvest or slaughter. Food spoilage can be defined as any change that renders food unfit for human consumption. Various factors, including contamination by microorganisms, infection by insects, or degradation by endogenous enzymes, are responsible for this. In addition, physical and chemical changes such as tearing of the plant or animal tissues or oxidation of certain constituents of food may promote spoilage. Any number of methods by which food is kept from spoilage after harvest is considered food preservation. The old methods, as we are all aware, include drying, fermentation, and refrigeration. In modern times, however, we are using canning, pasteurization, freezing, irradiation, and addition of chemicals. And our interest here is the irradiation. Radiation as a method of preserving food has several benefits. It slows down activities of disease-causing organisms, extends shelf life of food, prevents food from spoilage and waste, promotes trade in food commodities, and helps save money, reduce environmental impact, and contributes to more sustainable food system. Food irradiation is a process that uses irradiation to control microbes and insects in food to prevent spoilage. Food processing, an ally of food irradiation, is the transformation of agricultural products into food, or one form of food into another. Examples include grinding grain into flour, home cooking, or complex industrial processes used in making of convenient foods. So normally when we're trying to do irradiation, we must process the food first from one form to the other and package it before we do the irradiation, because after irradiation, you don't open it until it is ready to be consumed. So food irradiation works by exposing food products to ionizing radiation, and during the process, gamma rays, X-rays, or high-energy electrons pass through the food, destroying or inactivating microorganisms that cause foodborne diseases. The radiation process happens after a food is produced, processed, and packaged. Packaged food enters a chamber where it is exposed to specific amounts of radiation. The radiation penetrates the food and microorganisms and pests that cause spoilage. But the radiation does not make food radioactive. So I have here a diagram showing us what food irradiation can do. And this is the symbol for food irradiation. This single process I've explained on the right, and I've used the illustration on the left. Can be used to inhibit sprouting in our setting in crops like yam, maybe in onions, in ginger, and sweet potatoes. It can disinfest cereals and grains and flour. It can be used to extend the shelf life of any food commodity. It can destroy pathogens on our food, and it can be used as a quarantine measure that will enable you to export food across your boundary to other nations. So how have we applied this in Ghana? We have used it for disinfestation of cereals and pulses. Normally, producers will bring the material in bags like this, and then they do the packaging. Then we pass them. No. Scientists at the radiation center will pass through the irradiator and make sure the pathogens or insects are immobilized before the food is sent out for sale or for export. For phytosanitary and quarantine treatments in fruits and vegetables, on the right we are seeing boxes of fruits packaged for export. In a number of countries, you cannot send fresh fruits without irradiation. In the past, Binari had irradiated and stored yam for 6 to 8 months, and the product tested after the storage time was in perfect condition. Unfortunately, the focus here is not to bright. Otherwise, you have seen that the yam is perfectly white. There is no sprouting, and this is what the exporters want to see. Indeed, many have come to irradiate yam for export in the past, and I'm told that one of them once won the national best yam exporter. Other areas of application of food irradiation, we had a study which looked at extending the shelf life of fresh cuts, fruits under storage following irradiation. So there was irradiation, and the doses are as indicated. And then we also had, for comparison, the use of chemical preservatives, sucrose, citric acid, and sodium benzoate. And then a situation where we had a combination of these chemicals in equal proportions. Samples were stored at 6 degrees and 10 degrees for 15 days. And microbial contamination was assessed after the period. The conclusion after this is that irradiation is more effective and ideal compared to chemical preservatives in improving the microbial quality and therefore extending the shelf life of refrigerated mangoes. Mangoes that are cut and processed, put in a can, and you want to keep them on the shelf for some time. Okay? After 12 days, mangoes were still okay. And I thought this was an innovation that somebody could pick up to develop some big-time industry. Scientists also looked at pathogen reduction on ready-to-eat food, specifically jollof rice. And the doses of radiation applied were 0, 2, and 3, stored under refrigerator conditions, and the duration was 28 days. And the microbes were E. coli, Staphylococcus, and Salmonella. These are the observations and conclusions. A dose of 3 kilograms eliminated all the microbes from the jollof rice during 28-day storage period, and is therefore adequate to eliminate pathogens from jollof rice to enhance microbial activity. The study demonstrates the potential of irradiation to enhance microbiological safety and extend shelf life of ready-to-eat meats, ready-to-eat jollof rice. And once again, I thought this one too is innovative, because if you have a big restaurant, then you could allow workers to cook and pack the food and store in an ordinary refrigerator or some cold room at, yes, but nobody is doing this yet. The workers who did this, one of them is sitting here. We will move now to crop improvement using mutation breeding. And plant breeding is purposeful manipulation of the qualities of plants to create new varieties with a set of desired characteristics. In the traditional method, we select parent plants which have some economic use or have nice appeal to us, and we use control mating to mate these parents. And the progeny that come out of them, we select them based upon their performance. This leads to production of plants that adapt to human needs more efficiently, and we've used the method over time to improve quality and quantity of food for human and animals. The challenge that takes a long time, maybe often over 10 years. In the end, we don't create anything new, but we shuffle what you obtain from the parent plants. And for now, we are aware that we are not able to meet the demands of the challenges of our current time. These times we have climate change, we have population explosion, we have environmental degradation, we have increasing biotic and abiotic factors, all of which demand that we produce the new varieties now. We can't wait 10 years. So we've been looking for alternate methods to achieve this. And mutation breeding is a method of choice. It is a method that uses physical irradiation or chemical means to induce spontaneous genetic variation in plants to develop new crop varieties. And it involves the steps on the right, which I will not waste time going over because it will be boring to most people, but that's where we have been playing for several years. The benefits are that this method is very cost effective. It is quick. It has proven to work in all crops and over different environments. And it is transferable. If you do it on one crop, you can easily do it on the other. And this one, you end up creating something new that doesn't already exist in the environment. So, what have we done with this in Ghana so far? In 2014, Danso and colleagues did a review of achievements of mutation breeding in Ghana. And often the books will mention two main crops, cassava and cocoa. In cassava, the dry matter content was increased, and then the products became poundable. We are all from settings where once upon a time you cook your fufu and your soup is ready, but the cassava disappoints. Because maybe the variety is not good or the storage was not good, but this one was very good. There's also a record of resistance to cocoa swelling shoot, so we'll go over them briefly. Once again, I don't know where the projection is a bit unclear at the moment. But work on cassava involved a hybrid line obtained from Nigeria and then a breeder's line, and this was worked on in the IITA. And what the IITA does is it works on behalf of all West African countries, indeed Africa. So if they get something new, you can share with other African countries. So eventually this product came to Ghana, but it wasn't popular because the cassava will not easily make fufu. It's not poundable. So after irradiation, a new variety was obtained, which was named Tekbanchi, and had high dry matter content, 40%, and proved poundable. Stem cuttings were the materials that were irradiated. Indeed, when I came here to work as a young man, we had only a gamma cell. And even in that small gamma cell, we could put the stem cuttings for irradiation. So this is the summary from the researchers. So here now I put a comparison between the original parent and then the hybrid variety. Original parent was high yielding, the mutant also high yielding. Original parent resistant to cassava mosaic virus, same as the mutant. The original parent had moderate starch content. the mutant had high starch content. Original parents could not be pounded to make fufu, whereas the mutant was pounded up. So the increments were in two characteristics. Starch content went up, making it possible to get good fufu from this variety. Then I'll go to the cocoa. Cocoa, one of the problems is the cocoa swollen shoot disease. Good cocoa tree should bear pods like this. But the arrow is showing what a typical symptom of cocoa swollen shoot is. And that is a seedling with cocoa swollen shoot. And ultimately, infection by this virus leads to gradual dying of the branch or the whole plant. So, irradiation that was carried out here by staff of the Cocoa Research Institute working in tandem with Binary resulted in new varieties that are resistant to the cocoa swollen shoot. And now the Cocoa Research Institute can raise seedlings which are virtually free of cocoa swollen shoots. They have been able to cross this with their high yielding varieties. And now, if you buy your seedlings from them, you are sure to get something like I am showing on the right here. But I must add that if you are a layman, you may not get this. This is what they are getting with the help of the extension workers who are doing hand pollination. But the swollen shoot is almost out completely now. Also, some work on oil palm. I think that one was started some time ago, and it's stalled, and it's been resumed. And the nuts were irradiated in Ghana Atomic Energy here several years back, and they were planted at the Oil Palm Research Institute. So the partnership is between GAEC and IEA. GAEC and the Oil Palm Research Institute. It has been reactivated in recent years, and there is work going on. In fact, the idea now is to try and obtain as many variants from the mother plants which are standing. And one particular interest is to see whether we can get dwarf palms. I intended to show you on the left here palms which are in bearing, and then on the right palms which have become too tall. and putative mutants that were coming out of this have not been worked on since then, but we have potential to do that one. Then also some work was done on cassava, and these ones led to release of some varieties by the varietal release committee. Also some work by staff of University of Cape Coast involving cowpea, and these are the yield increases they got, and they even made some food products out of it and did a food fair. We've also been carrying out some work on soil water management, and basically using our neutron probes, we are able to detect how much minimum water some crops will need to give us high yield, how much fertilizer they will need to give us high yield, so that we don't waste fertilizer or we can produce under drought conditions. We call such varieties which can be so treated climate smart varieties, because if they are able to respond to just small amount of water, that is okay. Small amount of fertilizer, that is okay. Mr. Moderator, I think I have highlighted some of the work that Ghanaians have done. The main focus is the Ghana Atomic Energy Commission and BINARI. And we have shared partnership with a number of institutes across the country and beyond. Only part of the success stories have been displayed here. And I will encourage the director and current workers to have a plan of action for the next 10 years. We have a short-term plan, a medium-term plan, and invite all stakeholders and develop just a few flagship projects and get everybody working. The international collaboration we have done in the past must continue, and with local collaborators, but we must remember that if you are working in a vacuum, you will not get support. So we must align our work with national objectives. For some time now, MOFA has tried to project some 5 new crops, including mango, shea, butter, and other crops. And I think if we focus on those areas, and we'll be doing well, and be concerned about environmental sustainability, climate smart agriculture. And also, I think horticulture holds a lot of promise, because if you use irradiation on the flowers, you cause change. It's not like food that sometimes the plant will become stunted. Any change that you cause, that will bring something new on the market. You have hit it already, and you can easily export it. There have been problems with funding in the past, and I think they are still there. And so we must find new strategies of getting more funding to support the work. And sometimes it's good to recall institutional memory. So, and then the difficulty I faced preparing for this program is that there were virtually no documents. And then I think we must do a lot better in future. We used to have GAEC at a glance, BINARI at a glance. If we have something like that, just look at what work has been done in the past 30 years. And I think some of the work that have been done are good enough to go into commercialization. And Binari can benefit a lot from this. To get there, we have a socio-economics unit that can help people with ideas on how to start up businesses, move forward. But my last line is showing that you must be abreast with legal issues, because the guy who I'm told won the National Yam Exporter, after all that, tried to run away with what money was due Binari. And I think we must make them sign the necessary MOUs before we engage them. I think researchers in Ghana, with a focus on Binari and GAEC, have done extensive work in the application of nuclear science and technologies towards growth of Ghana's agriculture and food systems. Some of the research outputs are enough to be sent out for business development, and we must find time to link up with the appropriate end users. Of course, the IEA has already recognized this. And who am I to say that I can override what IEA said? It is already known now, and it was known long ago. The work of Cheche, which led to eradication in the north, prompted the IEA to donate a PIJO to GAEC for successful work in recognition of the efforts of the scientists. The work of the food scientists motivated the IEA to help build a radiation center. Today, plant breeding has been designated a center. We wish to come here 10 years from today to come and take part in the 40th anniversary. And we shall be happy to give you the applause you deserve. Thank you very much.
All the scientists in the institute. Thank you once again, Prof. So we are going to have the panel discussion, and I'm happy Prof brought this up because the lecture series for our 30th has not had any current scientists on the panel or presenting. And we recognize the importance of institutional memory. All the people who are presented from the first lecture to today and will be on the panel are people who are here from the inception of Binari, and we are not leaving them out of this celebration. And so it is good for us to interact with them and get to know the real story behind Binari and where they expect Binari to be. And I'm hoping 10 years time, Prof, you would honor our invitation to tell us if we've done well in the 10 years since your presentation. Right. I'm going to introduce our panelists and then we'll go into the panel discussion. The first panelist I want to introduce is Professor Daniel Kwesi Asare. He's an associate professor of soil science and has served in Binari for over 2 decades. During these years he was in Binari, he served in various positions which contributed immensely to the progress of the institute. Internationally, he has trained so many IEA-sponsored scientists and fellows from several countries, including Botswana, Kenya, Tanzania, Uganda, and Zimbabwe on nuclear applications in soil science. His area of interest includes modeling, agricultural water management, crop water use efficiency, fertilizer nitrogen use efficiency, and atmospheric nitrogen fixed by grain legumes. Professor Asare is currently a lecturer at the Department of Nuclear Agriculture and Radiation Processing at SNAS. He was my lecturer for biometry when I was doing my master's. And that was one course I was very scared of because I think all those who passed through his hands will tell you it's not an easy course to do, but he took us through and we are grateful for that. He's authored and co-authored several papers in peer-reviewed journals in his area. Ladies and gentlemen, please welcome with a round of applause Professor Asare. All right. So, to the next panelist. Our next panelist is Dr. Charles Emmanuel Ano. He's also an Odadier, and he's one of the very few Odadiers who went back to Presec to teach. And his students from the 80s still remember him. Whenever you mention GAEC, those who were in Presec in the 80s will say, oh, do you know Dr. Ano? Yes. He holds a PhD in entomology from the University of Cape Coast and has a rich experience in research and teaching for over 2 decades. And he was in Binari from 1988 to 2008. And during that time, he was a research scientist here. He was appointed to several committees here in GAEC, and he actually exited GAEC when he was head of department of the then Animal Science Department. Now it's known as the Radiation Technology and Pest Management Center. As a teacher, he taught at the Presbyterian Boys Secondary School at Legon, and he was also a senior lecturer involved in research and teaching of biological sciences at the Central He was part of researchers technology research project entitled Global Management and Conservation of Pollinators for Sustainable Agriculture through an Ecosystem Approach in Ghana, and this was funded by UNEP Jeff and FAO. Again, Dr. Ano was a project manager in charge of the mango ecosystem, and during that time he worked with farmers from Dodowa and the Yilo Kobo Mango Farmers Association. Apart from this, and I think you realized in Prof. Amwate's presentation, he was involved in so many other projects on sterile insect technology on the Cheche, and was very instrumental in the development of radiation-induced inherited sterility for the management of Lepidopterus maize stem borers in Ghana, among others. His research interest includes insects, pests, diseases, vector management on the mosquito, cheche, fruit fly, maize stem borers, and other insects of economic importance. Outside the world of science, Dr. Anor is a lay preacher, and he's a presbyter of the Presbyterian Church of Ghana, and is a professional counselor, and he's been registered with the Ghana Psychology Council. Ladies and gentlemen, please, with a round of applause, welcome Dr. Anor. And for many years, Dr. Anor was my neighbor, opposite us, flat B1. Prof. Amwate was my neighbor too, Bangalore, and Prof. Asari too. Right, our last panelist is Mr. Abraham Edujemfi. He holds a BSc Honors and MPhil in Biological Sciences for my department at Kwame Nkrumah University of Science and Technology. And he was appointed a research scientist in 1998 and served as manager of the Radiation Technology Center from 2011 to 2019 when he exited. And before becoming manager of the Radiation Technology Center, he was the head of department for the Department of Food Science and Radiation Processing from 2009 to 2011. He retired as a chief research scientist in 2017, from 2017, and retired in April 2019. He's currently at the Graduate School of Nuclear and Allied Sciences as a lecturer. He also serves as a consultant to Breforce Company Limited. Mr. Edujemfi has contributed immensely to the development of MPhil and PhD curricula, particularly at SNAS, and he has been a guest lecturer in economic botany at the Department of Botany, University of Ghana from 2006 to 2013, and served as senior lecturer for the Food Quality Management Program at the Department of Food Science and Technology, Institute of Distance Learning at Kwame Nkrumah University of Science and Technology. And he's been doing this from 2012 to date. In the area of radiation processing, he has served as an external examiner and has supervised quite a number of BSc and MPhil theses. The vision of Mr. Edujemfi is to provide relevant data and information through research and to accelerate the ultimate adoption of radiation processing technology for post-harvest management of agricultural produce and enhanced healthcare delivery. Ladies and gentlemen, our last panelist, Mr. Abraham Edujemfi. Hi, thank you very much, panelists, and I feel honored to be in your midst. We're going to have a discussion. Many of you have been here right from the days of the Department of Biology, Food, and what was the last one? Agriculture. And you transitioned into what we now know as binary. My first question to all of you, and to put a human face to scientists, is what motivated you to choose a career in the sciences, to become a scientist? I'll start with Prof. Amwate, and then we'll go all the way to Mr. Edujemfi.
Sorry. I went to secondary school as a small private student, and it was personally in my school that if you lose an exam, yes, it was one of them. So in my class there were 4 classes, A, B, C, D, and I was in the back of it. There were only 2 science classes, and then 1 for us, 1 for business. So by the time we continue the subject area of defined is competition, very stiff competition. I was fortunate to get an intelligent exam. And I believe it was also prestigious to do medicine. I wanted to do medicine. of the next those just in the young days. And I'll tell you, once I was a small student in the sec, we had one professor from he was Professor P, and he taught us physics. And his interest was more on the atomic physics. So he taught us GAEC for discussion. And that related my interest in learning GAEC when the opportunity came. Actually, I was director of school to teach. My senior was and stay here till I retire. Thank you. Yes, thank you. I know I've never wondered about this till today. He was very bright, very, very religious.
Thank you very much. Professor, why a career in science? Why science?
I think at the secondary school level, I was doing well, or I did well. So I decided to pursue it.
Okay. So unfortunately, you have to pass the mic when you're talking. They can't hear.
That's as a business. I did physics, chemistry, maths, and I was admitted at University of Ghana to read Agric. Agric, I realized that there were a lot of courses in biology, and I hated it. I hated the biology because remember at the A level, I didn't do biology. So the obvious thing for me is to either major in soil science or agri-economics. But I decided to major in soil science, specifically soil physics, because I had a good lecturer. He encouraged us to do it. So after the course, he introduced me to Ghana Atomic Energy Commission as where I could find employment. And luckily enough, I was employed. And I decided to take it up and continue with it. So that's all I can say about it. Yes.
All right. Thank you very much. Yes, so what motivated you? And I know you really love to teach. You taught at secondary school level, and when you left GAEC, you went back to teaching. So what motivated you to choose this path?
I'm very well my very happy I've been singing all the time. But in school, when you are interested in biology and all these things, you'll be aiming at doing medicine. So I remember when we got to section, we said medical society. All that was saying. But some of us didn't get it. But all the same, when I got to UCC, I did science. We were the first product student did a 4-year course. You do science and then you do deep education. That is, so you come out with your degree and then diploma in education. So I came to research. But whilst I was even there, I did my teaching practice 1 at research. So I came back to research and taught there. And even there, I realized I want to do my master's. So I did my master's. Immediately I had it, I said how to apply it to a research institution. I had CSR and atomic here, and by God's grace I got atomic and entered into the research field. And that's how I came to atomic energy and enjoying the research stuff.
What was your background in microbiology, and with microbiology, the world is at your feet. Why a career here?
Right from secondary school, biology was one of the subjects which really excited me, to use the word.
But when you hear me mic, it's most of the time.
I was really excited about biology whilst I was in secondary school. So those days too, I think it was prestigious to be reading science. So there was a lot of prestige with science subjects. So whilst at Opokuare, I really enjoyed biology a lot. And through, yes, I mentioned Opokuare, so I'm still reeling from the defeat anyway. So while I was at Opokuare, I really enjoyed biology, and I think I took it on to the university, KN University, while I read biological sciences. After that, I found myself teaching with the Ghana Education Service. I taught at a number of schools, and whilst I was teaching at TR Media, I really decided to move to the KN University and start a master's program. So I was doing my master's while I was teaching still at Media. And whilst there, I bumped into a gentleman who became a very good friend. And one day, I think when we were just about to end our program, I saw an advert in the papers, and it was about Ghana Atomic Energy. So I discussed with him, and he decided to come for the interview, and he was employed here. He happens to be Raf Ahiabu. So when I also completed, I was much in touch with him, and he told me about the place. So I also saw an interview, decided to apply. I think Madame was on the panel of the interview that day. I had a very, very tough time on the interview panel. Professor Fletcher, Abuasi, and the rest. But then I was able to get appointments. And since 1998, I've been working here. And I got in, and I also realized the area of food science and radiation processing offered so much excitement. There were so much areas where you could specialize in. So I got myself in radiation processing, got myself at the facility, and I think the rest is history.
And so please still keep the mic. And I like where you ended. The area of food irradiation is one that holds a lot of prospects. But in Ghana, when you talk about food irradiation, there's a bit of irradiation of food is safe. What safety features are in place?
Well, perceptions out there concerning the safety of the food. But radiation processing is one of the technologies that have been thoroughly researched over the years. And through these studies, that is consumed after radiation is safe. The first thing that I'll mention is the resources that are used for the purpose of radiation are carefully selected radionuclides. Cobalt and cesium are selected because of certain characteristics they have. For processing food, cobalt is preferred to cesium because cobalt is usually in a solid form. It is easier to handle compared to cesium, which usually occurs as a powder. So with this cobalt, the energy level of the cobalt radiation from the radiation from cobalt, that is the gamma rays, is such that the energy level is low and not high, and this prevents any chance of inducing radioactivity in the food. So one of the key things is about the energy level of the radiation, that is gamma radiation, that is used in processing food. Of course, we also have X-rays, which are also can be used in processing food, and there too the energy level is very low and is tightly regulated. We can also talk about X-rays, which are also used for processing food, and there too the energy level is also very low. So the first thing is all the radiations that are used in processing food have energy levels which are low and they don't pose any danger as far as inducing radioactivity in food. So that is the first point. Then the second issue is when radiation is carried out, the food is placed in a field of radiation generated by the radioisotopes. The food does not come into contact with the radionuclides, that is cobalt-60 and cesium-137. So it is rather the energy that is within these radiations that bring about the changes in the food. So that also assures us that there is no chance of any radioactivity being accidentally introduced into the food. Okay. So these are two clear instances noting that safety of food is very, very key as far as radiation processing is concerned. There are other safety measures also, but a lot of these safety measures are within the facilities where the food is processed through using radiation. So for now, I'll mention these are the two key measures that are put in place to ensure food does not accidentally become radioactive as a result of processing.
SIT has been found to be very environmentally friendly in the sense that it is target specific. If they say target specific, it means that the insect pests that you are aiming at, they are the direct pests that will be affected and not the others, because in an environment you have all kinds of insects. Some are pollinators, even some are even predators, which are doing biological, activities. So that is one thing that makes SIT very environmentally friendly. Apart from that, we try to ensure that the radiation that are given are sublethal, just like my colleague has said, gamma radiation, it's not very high, but again, when you study the particular pests, you have to know the biology of the particular pest, so that you can apply sublethal radiation that will not actually kill it. In fact, the whole idea is to, as we say, you are induced on sterility in the pest. You are not aiming at killing the pests, instantly per se. And so, normally the whole issue is also an integrated, approach of management. You use other control management. In fact, still, we cannot do without insecticides. They are there, but at a minimal level. So normally, SIT comes on board when the pests are almost at a very low level, and you want to reduce them drastically. Normally, when we say that something is a pest, that means they are above the economic level. So we also use other control measures to help us, to reduce the pests, and then SIT is to mob up almost, the pest activities in the environment.
The coconut beetle, but in his submission, he also mentioned that I think the larvae is of some nutritional benefits to those there. If we're going to use SIT, and I know SIT results in reduced populations, how do we balance reducing the populations and this?
Yeah, just like I was trying to say, in fact, we can't mix nature per se. Sometimes we say that we have extinct species in the environment. They may be extinct to some extent that maybe there has been poaching and so on. But really, we find out that one or two what we call remnants. The remnants will be there, but they will be at, below the pest level. See, when we talk about pests, well, we see that they become a nuisance. So if an insect is not offering any nuisance to the environment, it's not a pest. It's just like we are saying that we have some which are pollinators and so forth, right? So the whole idea is to reduce the pest activity, the nuisance activity. So I'm sure that coconut issue is very, very important because in fact in certain parts of the world, the larva of the beetle is a delicacy and very nutritious, yes. And in fact, a lot of insects larvae, are used as delicacies. Yeah. So you will not completely, kill them, I see, but your idea is to render them not to be pests. So they will be around and still they will not be a nuisance to you. But I understand even mosquito it's a pest bite, but it's not pest in certain meanings. Also, the way they have been, in terms of population. So, you can still make do with them. So that's the whole idea that we are saying. So we can go into it, and I think it can also help us to reduce the nuisance activity because it's very, very serious. This is, but it will not completely remove the harm, not just to reduce the nuisance.
Right. Thank you very much, Doc. To Prof. Asari, there's an issue in Ghana currently, and I'm sure you're very much aware. Our soils are being contaminated. I think just yesterday or 2 days ago, there was an article in one of the stations about high mercury poisoning of our soils. What methods do you think we can use to reclaim? I know I'm putting you on the spot, but what methods do you think we can use to reclaim some of these lands? Because whether we like it or not, they have already been polluted, and the pollution is happening in those areas where a lot of our food is being grown. What methods as a soil scientist would you recommend?
You are talking of contamination by metals, heavy metals. I would suggest that we should concentrate on using organic matter. The organic matter will mop up all the metals, or most of the metals, and they will not be released readily. So we need to make sure that we have adequate levels of organic matter. How can we do that? By applying compost, green manure, and the like. Another thing too is that even when we apply this in organic matter and we grow things, our plants will mop up the metals. If we have food crops, it means we cannot use the food crops. So we need to deviate from growing food crops on the contaminated soils. We should focus on growing forest edition trees, economic trees, like timber. You see, but not food crops. Not food crops. Yes, not food crops. Forest trees, timber crops, and they will mop up the organic, not organic, I mean metals. Yes. So as a follow-up,
in Ghana, anywhere you grow anything, it will grow. Is it true? If you say Ghanaian soils, you can grow anything anywhere and it will do well.
It's not true. It is not. Our soils are not generally fertile. Yes. Especially if you have sandy soils, like the coastal soils that you have around. No, they are not fertile. And then if you go to western region, where it rains a lot, the soils there are not fertile because of leaching of nutrients out of the soil profile. But the soils which are fertile in Ghana are those that have 2-to-1 clays. A typical example is Akuse series. You may not know Akuse series. You go to Akuse, a particular soil there. It's sticky. It's not all that workable during the rainy season, but it's rich. It's rich just because of the fact that there's minimal amount of leaching of nutrients out of it. It's rich. Yes. Apart from that, any other place, you need to work and ensure that you manage the soil so that the soil will become fertile. We should not forget. Don't say that the soil is okay, so it's good for me to use. No. Conscious effort to make it fertile. For example, the Akuse series. A major problem is the fact that it is sticky. When it rains, you can't work it. How do you go about it? Apply organic matter. Very, very important. Work organic matter into the soil. It will open it up. and then to take up a lot of water in the soil. It will allow free flow of air and water. And then we need to do that. Yes, in the north, to understand some of the soils too are not all that good. But as I said earlier, the application of organic matter and compost, we shouldn't go without them. Very, very important. Application of organic matter and compost will improve the fertility of the soil and will improve the physical properties of the soil. Remember, our plants, they need air, oxygen. If the soil is too compact, no, they will not grow well. It's only a couple of crops that will grow well when the soil is very compact. A typical example is rice and sugarcane. Yes, apart from these two soils, the soil should be workable, the soil should be porous for free flow of air and water. So that is it. Yeah.
Thank you very much.
You're welcome.
Let me come to Professor Mate. And I know this is your field, so I can throw it to you. How do you select and screen for mutant plants? Can you give us an idea how, when you irradiated in your screening for mutant plants, what are the considerations in there?
In mutation breeding, in fact, the event is a random event. So if you are irradiating the seeds or the stem cuttings, you don't know which of them will give you the desired mutant. So we deal in large numbers. You deal in large numbers, you have a higher chance of getting something changed to meet the taste you are expecting. So we irradiate large numbers like 3,000, 5,000, 6,000. So immediately after irradiation, you go and plant all in the field. Plant them such that each plant is standing separately, because you will go after they've come up to inspect each of them separately. And this is very, very laborious. So the first generation after irradiation is called the M1 generation in seed propagated plants, and vegetatively propagated plants is called M1V1. You go through laboriously with your technicians, inspect and pick out those that you think are still surviving but have changed a little bit and are showing the characters you are looking for. So you harvest each plant separately, and then for the next planting season, you go to the field. Those you don't think came up but didn't meet your expectation, just ignore them. Some may not even come up at all, they'll die because of the effect of the irradiation. So you move to a second generation, which is the M2 generation, and you plant the progeny of each harvest from a plant separately so that you don't mix them. These ones too have to be inspected one by one by one by one. Each time selecting those that are coming up with the characters you are interested in. And you do this for another generation and another generation. And the reason why you will do for 2 or 3 more generations is that in mutation, we are looking for a character that has changed and is inheritable. It means that in the progeny coming from the irradiated population, the character should be there. So if you don't go it 2 or 3 more times, maybe what you saw immediately after irradiation could be due to the environment or some insect infestation. So we'll do up to M3 or M4 before finally we select those which are coming up, have the characters of our desire, and also tag them. After that, then you are going to do trial of those that you have selected finally, maybe 5, maybe 10. You would do field trials to look at their expression of the traits you desire. We call it field trial. And you do that with a control, an irradiated material in mind. So of all that you have selected, whether 5 or 10 or 15 plants, some should be outperforming the others. And in the end, you will select maybe the best 2 or best 3. Then you move out of your environment and go and do what we call multi-locational trials to see whether, yes, they are doing well in other trials too. So the screening, yes, you have to have your objective in mind. I wanted a plant which is high yielding and disease resistant. In your field, the plants coming up, are they high yielding? Are they disease resistant? You must go and grow them. If you are interested in disease resistance, you must provide that condition in the field. If it is not in the field, you must do a greenhouse test first to see how they are behaving. But certainly, you must challenge them to the character you are interested in. When you are satisfied, then you select them. Thank you.
Thank you very much, Bob. So to start the next round from you, and then I'll come to our audience for questions. So this is an interesting question. I mean, people say that nature has everything we need. Why do scientists have to come in using nuclear science to cause mutations at all? Because for every mutation you cause, there could be a plant that exists in nature that is able to withstand or produce whatever you are looking for. So why do we as scientists have to come in with nuclear science to cause mutations in plants?
Okay. In nature, there is some form of mutation all the time. That is why there's mutation going on around us all the time. And in the plants, it's noticeable. That's why if you find a rose plant, horticultural crop, rose, you see that some of them have red flowers, white, pink, and yellow. They are all changes that took place over time. What we tend to do now is to accelerate that process, because if we want to wait for nature to bring up for us all the characters we desire in our lifetime, we may not get it. So what we are doing is to accelerate the process that nature has been doing. And of course, human population is increasing all the time. And the yields have become stagnant. So we must find a way of finding new plants which are yielding higher, new plants which are maturing earlier, because the window for rainfall has shortened from maybe 5 months to 3 months or 2 months. I must find a cowpea variety that will mature in 50 days instead of the 80 days. I must have cassava that will mature in 6 months instead of 1.5 years. I must have banana, which is short. You understand? So that when there is wind, it will not break. You understand? I must have cowpea, which doesn't shatter, because if it shatters in the field, I will lose everything. So these are what we look out for. Characters that will benefit us now, and benefit the farmer, benefit the end user, and are not available. We more or less try our luck. That's why we deal with large numbers. And once you find it, you hit it, you multiply, and you give to the end user. Thank you.
Thank you very much, Bob. Back to Professor. Using nuclear science in the soil. Can you explain to an ordinary person how you use nuclear science and technology for soil sciences?
Okay. Application of nuclear science in soils. Basically, we have used 3 different types in binary. The first one is the water management. The water management, where we use the nuclear science, we employ the neutron pool. We use it to assess soil moisture in situ at one particular place all the time. It helps us to, schedule irrigation. Apart from everything, we need that one. If you are able to measure soil moisture in situ at one particular place, you will be able to assess what? The changing soil moisture content in that soil profile. Based on that, using the water balance approach, you'll be able to estimate water use. Based on that, you'll be able to use it to schedule irrigation. And another area is the use of N15. N15 for nitrogen fertilizer use efficiency. The good thing about N15 is that it's not polluting. It is not polluting. It is not radioactive. You can easily trace it. So if you use it, you'll be able to measure accurately the nitrogen use efficiency by a crop. So that's the best method. If you don't use N15, I don't think you'll be able to get end use efficiency accurately. The other method will be something like the balance, which is not accurate. We normally call it apparent nitrogen use efficiency. But with N15, you'll be able to use it and get accurate nitrogen use efficiency by the crop. And then another area is the use of the same N15 to assess or to quantify atmospheric nitrogen used by legumes. That one is free. So we need to encourage that the plant is not going to pay. Well, the plant pays for it because the plant has to generate this carbohydrate in order to benefit from it, together with or in the presence of microorganisms. But it's free to us. It's not going to cost the farmer. It's not going to cost the government. It is free. So we need to be promoting that. In that case, it means we need to promote the growing of legumes in our farming systems, which is good. So with this, you can only achieve this through the application of nuclear techniques. So that is it. Without nuclear techniques, you cannot do it accurately. Forget it. Forget it. You can't do it accurately. But there's one thing that I want to tell you. For the neutral group, we have to be very careful with it. It's dangerous because of the radiation in the neutrons. So normally RPI controls it. We are given badges, TLD badges, and periodically they come and collect the badges and assess the dose that you have received. If it's too much, they have to don't use the equipment for some time. So you know that because of this, it's not anybody who can use neutral group. You have to be trained on it. But it's very effective. It is the single most effective method for measuring soil moisture content. Other methods are come, but the neutral probe is the most effective method, accurate method for measuring soil moisture content. Yes, this is what I can tell you. Yeah.
Thank you. Thank you very much.
Yeah.
Thank you very much, Prof. So, Dr. Ano, a couple of years ago, maize farmers faced a huge challenge with the fall armyworm. Being someone who was part of the research that has now resulted in a reduced number of the Cheche control program, what advice would you give the government to try to use nuclear science to control this very devastating worm? What advice?
Right. I think when Professor Wante was doing his summary, he also mentioned that started some work under pest management, also use biological foods and other forms. One thing with this armware is that it's lepidopterus, and that's what I've said. You have to study the biology of particular species. And they are also radiation resistant. So again, you study all these things and then you try to know the dose, some meter dose that you give. And so in the lepidopterans, what we do is that we have what is known as radiation-induced inheritance sterility. That's also a form of SIT. SIT is a broad thing, but radiation-induced inheritance sterility is you are aiming at offering that supplemental dose so that the F1 generation, that is the parents offer the supplemental dose, and then it will reduce, the fecundity, but the F1, which is the first generation, you find out that if you mix them with the normal, you have a reduction. So again, it's not as easy as just going to it. These are some of the techniques that you have to do so that you do it. And again, you do a lot of the things in the lab. Then you also do something in the cage to find out how it's working before you get onto the area where again, now we are coming on the need to get involved in area-wide pest management in terms of SIT, because if you do it only in Ghana, you realize that by the time you have done some population reduction here, they will also get out from other places to come and invade the place. So some of these things, if you are serious about it, we have to get into maybe the sub-region that are thinking about, the ECOWAS region, that is West Africa, and get the countries involved, because it's not only in Ghana that we have having resources, but the whole continent. But we can start somewhere from West Africa and East Africa. So again, it's a long process, but we'll get somewhere. So long as we have the commitments and we have the political and all these things, I think if we give the resources, the researchers are there, deliver, and we need the resources.
All right, thank you very much. And my last question before I come to the audience. To Mr. Dijemfi, we've done a lot of work on gamma radiation of food and the use of gamma radiation in the agricultural systems of Ghana. What do you think is hampering the commercial uptake of this very important technology? Gamma radiation is good for food preservation, for mutation breeding, and for all the other things. But in Ghana, we seem not to be ready. What is the challenge? What, in your view, is hampering commercial uptake of this technology?
Yeah, thank you for this question. I think over the years, the country has gone the path of trying to build capacity in the area of food irrigation. This has taken us through the establishment of the facility that is currently on site. We've done a lot of work. We've done other studies. We did a semi-commercial work as far as the facility is concerned. But now we still have challenges. And I think commercialization of the technology challenge that is facing us as of now. The public, number one, has a perception about nuclear technologies. And I think we mentioned the issue of safety and the perception of the activity of food that is irradiated. So there is that public perception out there which always is working against adoption of the technology fully at a commercial level. So that is one reason. There are other reasons, and that will also be the difficulties in transitioning from semi-commercial to fully commercial operations as far as the technology is concerned. Our example here at Atomic Energy clearly shows that to transition to a fully commercial facility is really a challenging one. You need to get a facility which is up and doing. And our experiences have always shown that anytime we try to build the stakeholder base to keep using the facility, our setup becomes a bit inefficient. In other words, the technical aspect of getting the right source in terms of the cobalt 60, getting the right strength over a long period of time, so that the initial stakeholders who start bringing their produce will continue to come, because you need to build the market. And the experience we've had is whilst building the market, we've always had a problem with sustaining the source strength. And that always has been a challenge. Most of the stakeholders get a bit disillusioned about how they can continue using the facility when efficiency is going down. So the issue of getting the facility to be in operation for a relatively long time, to be able to sustain the interest of the key stakeholders, meaning the food producers, the exporters, so that they can continue to rely on the source. So the challenges of maintaining the source value is very key. And also we have this public perception about people who we also want to find excuses or reasons why they want to irradiate their products. But these are the bulk of them. We can go into details and I think get more subjects as to why this is still persistent.
All right, thank you very much, Mr. GMP. I'll throw the question time to us and the audience. Anyone with questions for, and you can see the depth of knowledge that they have. These are people who have been in this field, and even though they've been retired from the commission for a while, they're telling you how knowledgeable they are in the field. So, any questions? Yes.
I would like to thank you for the presentation. It will be good for me to ask every panel member a question back. I'm not the only one here, so I just go to Professor Sarah. So what do you recommend? You recommend that farmers come for testing before they start their farm, soil testing. Do you recommend? Can they go for soil testing before starting production? Thank you.
Having worked here for many years, I have known our facilities and what we have, and I what we can do. Prof. Mate, I've also known you as a farmer, mango farmer. In fact, I've eaten from your farm before. My question is, what would you recommend to your colleague farmers to take an advantage of what we have, or you yourself? I mean, having been with us for some time now, considering our facilities and capabilities. So at least it will not just be a mere research, and also, extending our hands to the farmers on the field. To Dr. Chasano, my question is, if you have to concentrate our efforts on the variety, since you indicated it's vector specific, will you advise that since it's a vector or insect parasite, we get it totally like extended from our system, like they should not leave since they remain vectors or insect parasite if we are using the SIT, which is vector specific? Thank you.
I want to thank all our panels for their contributions. I have like 3 questions. And my first question goes to all the panels. My question is, I think the basic aim of science is to improve the standard of living and make life easy. So I want to know if there are any measures that we are putting in place to attract individual investors and entrepreneurs so that it can improve their interest in the agro-business sector. Since there's a lot of unemployment in Ghana, and that's one of the major problems, is there any measure that are in place to help the youth grow their interest in the field? Because in Ghana, I think a lot of youth don't have interest in this sector, but it's a very lucrative business, so that is one of my questions. And my second question goes to Prof. Daniel Asari. He made mention of the neutron pool to improve soil moisture. This means that the workers in this field have a little health risk. I want to know if there is any form of insurance policy or motivation for these workers in order to encourage others to venture in the field and not scare them away. And my last question also goes to all of the panels. Should we believe that the nutritional value of the fruits irradiated are maintained, and that the ordinary Ghanaian can have a sane mind to consume this product? Example, the mango mentioned. Thank you.
Quite a number of questions, about 5 and so 5 or 6, depending on how you break it up. So majority went to Professor Sari, so you have to go first. A question. Stella.
You want to know whether definitely soil testing is very important, and it is required. However, no, but it is expensive for our local farmers. But if you are engaged in large-scale farming, please, you need soil testing, because without it, you can't get any loan from the banks. It's very, very important. The bank will request it, that you have soil testing, and you bring the report to them, because we are going to engage in a large-scale farming. So you need to devise strategies to manage the soil effectively. So it's only soil testing that you can use to do that. So large-scale farming, soil testing is required, whether you like it or not. Thank you very much.
So, you see, individual farmers may find it difficult to do that. Do you recommend, I know that there are steps now by government to have a soil map of Ghana. Do you recommend that they speed it up so that this can be made available to farmers, so that I know I can grow maybe coconut in this area, I can grow mango in this area, I can grow cassava in this area?
Fidelis, for individual farmers, even commercial farmers, if you are hard up, get in touch with Soil Research Institute. They have all the maps, capability maps. They will be able to tell you where to grow which crop. So do that. But for an individual like this farmer, local farmer here, you can talk to one of us. You can talk to one of us. We'll come to your field and we see one or two things. We'll give you a lead. That, okay, you need to do this, you need to do that. Even the vegetation on the field, the weeds on the field will tell me whether the soil is fertile or not. So if you don't know, please talk to people. They will guide you. Yeah, the lady wanted to know the neutron probe. Okay, I would say insurance policy, but you cannot use neutron probe without RPI. Radiation Protection Institute. You cannot use the neutron probe without Radiation Protection Institute. Radiation Protection Institute will have to register you. They have to know where the equipment is, who is using it. Has the person been trained? Yes, and then they will give you a badge, TLD badge, to monitor you. Periodically they will come. Yes, collect the badge, do the analysis, and inform you as to whether it's okay to continue or to stop you from using it. Yes, it cannot be used by any untrained person. No. Neutron probe, the person to use it must be trained, must know what he is doing. That is it. Yes, my lady, you wanted to know. I don't know whether the insurance has something, but you just get in touch with RPI. RPI will guide you. But the quick guide, if you are using it, don't get closer to the instrument all the time. For example, this is where I'm measuring. I have the instrument mounted. As soon as I have the instrument mounted, and I've started operating it, I move out. When the instrument finishes reading, it will give me a beep. Then quickly I go and read. You see it. So don't get closer to the distance. And I will also advise that if you are pregnant, a woman, please advise yourself. Yes. It's a fact. It doesn't mean that it's very scary. No. Even the radiations that you receive in a plane is more than what you receive from the instrument. But all the same, protect yourself. That's what we tell you. All the same, protect yourself. Thank you very much. As somebody was saying, what can we do to assist the youth? What? To attract into nuclear science.
Yes.
Oh, can somebody
Especially agric and the nuclear techniques that are used in agric and food systems.
Thank you very much. I'll start with the gentleman, the is asking why we have generated so much technology and the farmers and users are not coming after us. Okay? I will give you personal experience. For 3 years of my working life here, I took steady leave to work with a group of pineapple farmers. And my experience working with them taught me many things. The people I worked with, pineapple farmers, they were once government workers, self-made men. In fact, one person had an aircraft for lifting his pineapples from Ghana to Europe. They were businessmen. Businessmen want solutions today. They don't need your solution tomorrow. So if you want them to take up your innovations, you have to go after them. You understand? Yes, I am into mango, but if I have to wait for Binary to come and do the area-wide control of fruit flies, it means 3-4 years I can't sell anything. So, instant solution, I'll buy chemical and control the fruit flies on my mango. You've been seeing the people selling the, what do you call it? Items they have a vehicle, they put their music on it, and they are selling. And they are selling their produce. They are bringing it to you. So if we want them to take up our technologies or our innovations, we must go out to them. Unfortunately, in government budgeting, we don't have money to do that, and that is a major headache. And in my presentation, I tried to say that, well, we must adopt a new strategy now, and that from now on, we must do what is called demand-driven research. You understand? Demand-driven. We had the experience some time ago. The same pineapple farmers, they destroyed the market for smooth cayenne, which originates in West Africa here. And they got another variety called MD2, and they wanted to produce this. People had 200 acres of farm. People had 2,000 acres of farm. Where were they going to get the suckers to plant? And they got to know that we had a facility here, and they came. We sat down with them, and we produced for them. If you can produce for them instantly, they'll come back. You understand? But if you ask them to wait 6 months, 1 year, you are not giving them solution, they won't come again. You understand? So if you have a solution, you can go out to them. I think the food irradiation people should go to Ghana Standards Authority or the Food and Drugs Authority. There are people willing to export products, but the contamination, the storage and things, but you tell them that if we irradiate it for you this way, your goods will go there and they are still good on the shelves for 3 months, 6 months. We must sell ourselves more. Thank you on that one. The lady is saying that how to attract individuals into S&T usage. It's the same thing. If we are able to demonstrate to them the importance of using nuclear science and technology to overcome their problems in their production practices, it will be fine. Several years ago, I think Professor Mills was the president at that time. We did the YAM, large-scale irradiation. We did a food fair, like Food Research did a food fair weeks back. And we invited all the people who matter in Ghana, Minister of Agri and everything. And the First Lady, Mrs. Nadu Mills, was the guest speaker. And the food fair was held in the court here. And everybody came, and they ate everything, and they got convinced. And based upon that, a few of the exporters came to irradiate their things to export. So I think we must go along those lines. When we find an innovation, we must do what we call in our place it will be called Farmers' Day in plant breeding. You bring them, demonstrate, let them see. If they are convinced, they will take it up. What else is the other one?
So there's a question for whether we should completely eradicate
let me try some more, then I'll give it to the entomologist. You know, we live in association with all organisms, and we shouldn't be selfish and say that we want to eliminate this one or the other. In the ecosystem, every organism has a role to play. So in pest control in general, the desirable portion is to reduce the population of the pest below economic level, below the level that it will cause economic damage to your produce. So managing the pest population is better than eliminating it completely. Thank you.
Yeah, so on that same issue, like I want to add that a lot of people have certain perception about SIT because you do mass rearing and then you release. And I said earlier on that it all depends on the specific, pests. You don't do it for all the pests, yeah. And in fact, the father of SIT, it's Edward Niplin, who brought the idea some years back in the 30s, 1930s and 50s. And it was first started on this new word, screwworm. Screwworm, they are a type of flies, that feed on the wounds of, cattle, horses, and so forth. Yes, and they grow very widely, and maybe they may even kill the horses and so forth. So it was started in the US. And you realize that the screwworm, for example, just like the fly, okay, when the female receives the sperm, it blocks the entrance so that you will not have another male coming in. No, they store, we have spermatophore, they store the sperm in some special, organ in the body. It's not for all insects, okay? So when you study that, so that's how it happened. And when he used the SIT for the first time, he realized that they have to produce millions of the male, because the male that will, mate, and then they will also overwhelm the normal in the natural population, so that they would then attack the female and then, getting the sterile sperm. Do you understand? So eventually you realize that they will not reproducing the fresh, flies. That is the screwworm, okay? And it was very effective. They were able to, almost, eradicate that problem. It forms what is known as myiasis. Myiasis is the disease. And it also happened in Libya, and it was tried there. It also, stopped everything. So that's how the SIT started. And so now we also have what is known as you trap it, and then, you trap the insects from the environment, then you radiate it, and then release. So now it's the trapping, radiates, and release. So you find out that when something starts, as we go ahead, we begin to modify the whole thing. But the basic thing, it's the genetically modified issue that, you want to occur. So that, it's something like, maybe you will say, a human being, what do you call it? The way we use all kinds of this thing to reduce population. I mean, you use pills and so forth in the female and so forth. Yeah, so it's a way of trying to reduce the population. And now we are trying to you trap it within the environment itself. So now it may not be necessary even to mass rear. And that also depends on, again, the particular, pests. We are trying it on the fruit flies. They have started some work like that. And just like Harry said earlier on, see, a lot of these farmers, they want things quick. But, research takes a long time. And again, that's why we do integrated, pest management. So you'll be adding other things that are also environmentally friendly before you get that the air sighting to come on board. And one thing I also want us to encourage ourselves, we have to, just like one of the ladies was asking how to motivate the researchers and so forth, I think we have to make some noise. Recently, the Gabonese president who was overthrown, he said, make noise, make noise, make noise. And when he was making noise, they heard him. They heard him. And then, so please, let us make noise. The researchers, we don't make noise. You see, let us write up short this thing into graphic, into newspapers, just like he was saying, when we organized some food fairs some time ago. And when the people from outside came and they tasted some of these things, they realized that, yes, there's something good here. So, if you don't make noise about yourself, how? Hallelujah. So, thank you very much. That is what I want to add. Do you have a word to say?
Yes, from the lady. She said, is the nutritional value of fruits irradiated maintained, and can the ordinary Ghanaian have the same to consume this product? Example, mango. Is the nutritional value the same? Is it affected by irradiation?
The same. If you look at food and irradiation, I think one of the first things that was settled was whether irradiation, aside the benefit we get in terms of maybe destroying insects, in terms of destroying microbes and other things, the food itself is not being impacted by the effect of the radiation. But this has been proved not to be so. There is no evidence indicating that irradiated food suffers from nutritional defects. A lot of work has been done over the years. I think through feeding trials in most of the countries which pioneered the technology, China, US, Europe, and feeding trials were undertaken in laboratory animals and also in humans. And there is no evidence that food that is irradiated becomes nutritionally deficient. What we know is that some components may be affected in terms of the dose that is applied. For example, if you are looking at fruits and vitamin C, the dose may affect the vitamin C in terms of reducing the level of vitamin C. But there are ways of trying to compensate for this or reducing these effects. Where there are no ways, the products may not be recommended for irradiation. Okay. Aside vitamins, I think all the other components, minor changes, but they are not significant. Yeah, so the conclusion is when food is irradiated, nutritional inadequacy does not really come in. Yeah, and that is what the scientific expert committee has actually stated. Yeah.
Thank you very much. We want to have their last word and then we call it a day. Your last word.
My last word?
Yes.
Okay.
Let's go around the table. The last word. One last word for us. Yeah, I know Opoku are lost, but just one last word.
Yeah, what I would say is the area of food irradiation as a component under the nuclear science and technology applications is a very important one, but the country as a whole is not getting the full benefits of this technology. And going forward, I will wish that the area or that sector gets full support from governments. or get some assistance from government. We've been able to demonstrate the effectiveness of the technology as far as preserving our staple foods are concerned, as far as reducing post-harvest losses are concerned. The technology has gone through the level of pilot scaling. Now we are at a stage where we have to go into full-scale commercial processing. And there is a need to get some support, either materially or in kind, or maybe in terms of financial support, to make this last transition. And until that is done, all the investment that has been put into the sector will not be realized in terms of benefit for the country. So it is my wish that going forward, the sector gets the needed support to enable us to assist in the socio-economic development of the country. I am aware that efforts are ongoing to secure funding from government to upgrade the current facility and also to get some assistance to set up new facilities in other growing centers in the country, especially the food producing centers of this country. It is important that these efforts are sustained so that the country can reap the benefit of the long-term investment that we've made into the area of food irrigation. So on this note, I want to remain optimistic, knowing that the department, Binari, has the right requisite of talent and expertise to ensure that this venture succeeds for us as an institute, for us as a commission, and also for us as a country, that nuclear science and technology can be a savior. It can help us to resolve our food challenges in future. Thank you.
Yeah, so again, like I said, science research, it is about your findings, but we have done a lot, which so many people don't know. So let us make noise, make noise, make noise. The little one we have heard, we have done, let us make noise. They will hear us, and then they will help us. Thank you.
What can I tell you? Well, some people have the notion that nuclear science is something different from the actual science that we know. But I want to let you know that you cannot do nuclear science without the regular science. They go hand in hand. They go hand in hand. The nuclear science is superimposing something new, something different on the regular science. And then one thing too that we need to remember is that nuclear science is expensive. Nuclear science is expensive. If you are going to use it, it means it has some advantages. Typical advantage is that it could be fast, give you accurate results. Yes. And then it will be cost effective eventually. Initial cost will be high. N15, 500 grams of N15. Those days, it was between 1,000 and 1,005 US dollars. So it's not cheap. It is not cheap. You must know what you are going to do. If you don't know what you are going to do, please don't apply nuclear science. Thank you very much.
I will comment on two issues. I was And all those years, we faced problems with funding. So, Mr. Director, I think we must change the strategy a little bit. We must work on fewer projects and get all hands on deck. We must identify flagship projects and form teams to work on the projects. Scientists is either writing proposal looking for money, working in his lab or field, executing that project, or is come into town looking for collaborators and end users who will come and take up the end products and bring some money to Binari. Government funding is almost but sometimes too, you must work your way to the ministers and sometimes the chief of staff, I remember years before. There was one time I went to my director. I went to the office of the Minister of Agriculture till 8 p.m. You understand? He asked us to come back to the point. I said, we were patient enough. That's when everybody has gone home and he's not doing any of his usual things and he can listen to you. You understand? But of course, when you are going, it's a little bit of protocol. For those days, I had a lot of pineapple. Anytime we're going, we'll take some pineapple or plantain or things along. It works everywhere. If you are going to the chief's house, you carry some snap. So if you are going, you are lucky the chief director is here. So, but if you are going to other chief directors, you must take something along. I beg you. You understand? Yes. And as for the workers, I think it must be more firm on them. Those who don't want to work, they must be flashed out. You understand? Okay. The other thing that worries me is that we have a lot of people working in the area of nutrition. But if you go to the international circles, apart from the cocoa and cassava I talked about, nothing is documented in the books of the IAEA because we have not met their standard. If you think you have generated a mutant, let that mutant be validated and confirmed by the IAEA. Then your product will go into the IAEA database, which has all the mutants. Now, if you check Ghana, some of the documentation is Ghana zero. You understand? So for a number of those crops I have mentioned, they think they have found mutants, but nobody has confirmed. In the olden days, the IAEA experts used to come. In the case of cocoa and cassava, I know, but they came to inspect the product used here, and they were convinced that they were true mutants. These days, you don't even need that. You can do small molecular biology tests to confirm that yes, something genetically has changed, and send your result to the IAEA. If they are convinced, they will document your stuff in the IAEA database. So I think we must pursue this one to ensure that we tell Ghana, Africa, and the world that we are also doing something. We are doing it, they know, but if you don't use the right channel, then it's like we are not doing it. Of course, let me use an analogy. A lot of footballers parade themselves as good footballers, but you must go to the international scene and be tested and confirm that you are an expert before you accepted as such. Thank you.
Thank you very much. I think there's very little for me to say at this moment, and to say thank you to the panelists. It's been a pleasure to moderate this session. I'm looking forward to moderating you again in 10 years' time. And when Binari is 50, I may be joining you on the other side. So maybe I may not be a moderator, I may become a panelist then. Thank you very much, and thank you to the audience for staying with us. We've gone beyond time, but you're still here with us, and we are grateful for coming. I'll hand over to the MC. Thank you.
you are released to go back to So I will walk to one person who randomly, you tell me your take home from today's public lecture. I'm sorry if I come to you, I don't really mean to bother you, but it's just a random face, random. Please, your take home from today's public lecture. No, one person, because a lot of people were dozing. I was seeing that. So your take home, you did. Hey, so all the things that we were told here, nothing. Because time, okay. You are just stepping in for them. So if you're not able to answer, 50 Ghana each. So when we close, I'll collect it. So we'll take our closing remarks from our director of the institute, Dr. Assign, please.
Thank you very much to everybody. Thanks to our keynote speaker and to the panelists for doing justice to today's topic. I think a lot of us have learned a lot. Myself, I thought I knew all about nuclear applications in agriculture, but I have learned so much today. It's always good to, people say, it is in the old palm tree that you have a lot of palm wine. Yes, and they have demonstrated that experience really teaches so much. And today we have tapped from their experience. And for me, I'm privileged that the people who were there at the beginning are still here with us. So we can tell the full story of Binari before they leave us. So we will continue to come back to you. We are writing the history of Binari. We'll put all these pieces together, come back to you to validate some of the issues and the things that we are putting in. So we have a very concrete story to tell. And as the moderator said, 10 years from today, we will meet again and continue to tell our story. I am also very challenged by some of the issues that have been raised here and the challenges that have been posed to myself and my team by the keynote speaker and some of the questions that have come up here. We are doing so much, but very little is known about us. Very little is known about what impact we are making. Even within GAEC community, talk less of across the country and globally. So there is a lot of work for us to do. And my consolation or my hope is that I am young and I have a very young team of scientists, very vibrant scientists, and I'll call on all scientists in Binari to let's join hands together. Let all of us put our hands on the wheel and we can make greater impact so that in 10 years' time, when we sit here or we come here to tell the story will be nicer and we'll be more proud about what we have achieved than what we are seeing today. On that note, I want to thank everybody and all of you for your support. We want to thank the people who have really supported our work over the 30 years, including the international partners like IITA, IAEA, ICIPE, CABI, IFPRI, and all the international agencies that have been supporting our work. We also are very grateful to GAEC for allowing Binari to grow within this space. If GAEC hasn't given us the opportunity to grow, we wouldn't have grown from Department of Biology and agriculture to what we are today. And then to all of you who have been working hard behind the scenes, thank you very much. We thank our board chairman and board members for making time to join us in the meeting. They were here before all of us came. And we have kept them here even beyond the time we are supposed to close. Thank you very much for your time. And we are very much assured that you are in for us and you are in for our good. And we will keep working with you to make Binari great. We want to thank the organizers of the program, led by my Deputy Director, Dr. Oklu, for all the effort that you have put in to make this public lecture a success. We want to invite you to the climax of our 30th anniversary celebration, which I said tentatively is fixed for 24th of November. If there is any change, we will communicate that to you. But we are hopeful that we can have the event on that day. And we have a lot in stock on that day. As one of the speakers said, they had a food fair. We're going to have a food fair on that day and demonstrate all the different things that Binari has been producing. People will taste, and you can buy some and take away, and all that. I want to say a big thank you to Director General for staying here with us throughout the program, and then the Deputy Director General and all the directors who have been here. Thank you very much for joining us and being with us. Thank you.
Thank you so much. Please, before we close, I think it is very right that we take a short word from our Director General. Since Director General is here, just a calming words will do. Thank you. Please give it up to him.
Asamaba. Thank you very much. First, let me appreciate the panelists and the keynote speaker for what they've done. Even this morning, I was doing Binari's bid for them. We had some visitors from US, the Nuclear Regulatory Commission. So when we're taking them through what GAEC does, then I mentioned gamma irradiation facility, and was telling them how we can use the gamma irradiation facility to deal with post-harvest losses. And at the same time, we are talking about recycling of plastics. And I said that we can use irradiation facility to deal with this plastic pollution menace and all that. So, Binari, I did something for you this morning. It's been very, I know Binari has very huge potential. Huge potential. Anytime I meet with people, they tell me, oh, Binari is there. And I can see that they need our support. And I know my administration will do that. We'll do our best to support GAEC. We have low-hanging fruits which we can easily tap and change things around. And we're going to do that. And we're also happy to have a chief director who is so much in love with GAEC. Chief Patrick Nomo is here, and Chief will support us. He's already demonstrated that we needed some money to do something. We needed 80,000 euros from Ministry of Finance to support us at Aqua Thermal Hydraulic Loop for the school and for the training of nuclear engineers and others. And Chief assisted us within a short time to get the money to us. Chief, we are grateful. So, as I said, we've learned a lot about what we can use nuclear science and technology to do in agriculture and food systems. Let's carry this message to the general public. People ask, what do you do here and all that, but we can tell them that we can use. We are grateful for this collaborative center status that has come to GAEC. That is first of its kind in Africa, and we are so proud of you. And we have a lot to do because the director of Division of Africa would also like to meet with Binari on that day to discuss the modalities involved, the steps that we're going to do, the projects and the program that we want to implement at the collaborative center. No, it's this Friday, yes, 3rd November. It's talking about the public lecture. It's this Friday. Thank you very much for prompting. So on this note, I want to thank you all, even though they've already thanked you. Let me also add my voice. So thank you all for making time to be here. And we've enjoyed ourselves. We've learned a lot about binary and how we can use our nuclear technique to help Ghana solve our problem. Once again, thank you.
Thank you for thanking us. So we'll take announcements from the Deputy Director of Binari.
So just a short one. After the program, we'd like to meet the panel members and board members at the Binary Directorate, Binary Boardroom, where we close. Panel members and board members would like to meet you at Binary Directorate, the Binary Boardroom. Thank you all for coming. Make sure you have something before you leave here. They are distributing. Make sure you have that. I think the next thing will be announced by the MC. I think it's photograph. Yeah.
Thank you very much. All too soon, the program has ended. And just as we started with a prayer, we'll end with a prayer. And please, after the closing prayer, we'll move out to the podium there so we can have a photograph. The room is quite dark, coming from the media team, so they want us to take the photographs outside. So please, let's all be part of the photograph. Thank you so much. Thank you everyone for coming. Mr. Khalid Kusi, can you please give us the closing prayer?
Okay, shall we pray? You say, Alhamdulillah, that we give thanks to Almighty Allah for giving us this successful program. And we also ask Allah to ease our challenges and all our programs that we have for the coming months and coming years, that it gives us a befitting fund for those prayers. Alhamdulillahi Rabbil Alameen, Ar-Rahman Ar-Rahim, Maliki Yawmiddin, Iyaka na'budu wa iyaka nasta'in, Idina Siratal Mustaqim, Siratal Lazina an'amta alayhim, Ghayr Maghdubi alayhim waladdallin.